Good afternoon. I'm Eliana Rajani, and thank you for joining us today for the Science Speaker Series. Our guest speaker, Sarah Carter, will be providing insights into the world of drug development and clinical trials, describing her daily work life as a development executive for a pharmaceutical company. Ms. Carter attended DeMontefort University in Leicester, England, and received a dual degree of Bachelor of Science with honors in Biomedical Sciences and Chemistry. She later attended the University of Wales for a master's in clinical research with distinction. Ms. Carter started her career working in a laboratory for the National Blood Transfusion Service in England. After 3 years, Ms. Carter was fortunate to gain an entry-level role as a clinical research associate that began her career in drug development and clinical trials. Ms. Carter has experience in the delivery of clinical trials across several indications, holding different positions of increasing responsibility. Previously, as the head of global studies at Amgen, she managed all aspects of clinical site startup and supplier performance across over 1,000 staff members. Ms. Carter was the leader of a large, diverse organization, designing innovative ways to increase the speed of development programs, as well as providing the tools and knowledge to allow clinical study teams to deliver the results of their clinical trials. After 17 years at Amgen, it was time for a change. Currently, she is Vice President of Business Operations at Nuco Oncology Therapeutics Company. This growing biotech startup in Newbury Park specializes in small molecule oncology therapeutics. Ms. Carter's role is to ensure the smooth running of all aspects of the company and to collaborate with researchers. For today's Science Speakers event, we will be allotting 25 minutes for Ms. Carter to answer designated questions, and afterward you may ask her a question in the chat box. Thank you for watching, and we look forward to today's interview for the Science Speakers Series. Ms. Carter, thank you so much for joining us today. Let's begin with our first question. Please describe your education in both undergraduate and graduate studies and how it led you to your current field. How did you decide to pursue this field?
Thank you for having me today, and I'm excited to talk to everyone. So from the minute I speak, you'll realise that my undergraduate and graduate education was not probably the same as you'll experience here in the US, as I actually went to school in the UK. So it's slightly different in the UK. At a much earlier age, you specialise, you decide what you're interested in. And I think that was key to me in how I ended up in my career. So at the age of 16, you choose 3 to 4 subjects that you study in a really deep sort of focused way. So at that point, it's hard at 16 to know exactly what you want to do. So I chose what I was interested in. And I'd always been really interested in biology. But I hated plant biology. Plant biology was just not my thing. And human physiology and the way that the body works and how things react and how you put it all together, that was what was really fascinating for me. So my first sort of thought is I chose what interested me and what I knew I could remain interested in. And I took that forward. And when I applied to, we call going to college, going to university. So when I was 18, I applied to go to university. I kept that same sort of interest. So I started studying biomedical sciences with chemistry at the same time. One of the key things that I was lucky enough to do in the UK, you do a 3-year course to get your Bachelor of Science, but you can add in a year where you go and work. So I decided that I'd do that and go and get some more experience before I took my final year. And so I headed off to Cambridge, and I was lucky enough to get a job at the Cambridge Blood Transfusion Service. So I was able to work for 12 months there. And I decided that working in a lab was really interesting, but actually also quite routine. I graduated from my BSc, but then I went back because if you're sort of 21 and you have a job, you tend to want to go off and do that job. So I went back to Cambridge and I remained what was called a medical laboratory scientific officer. And transfusion science is kind of a part hematology, but part genetics. People hear about blood groups, you hear about A, B, O, AB, positive, negative, but there's actually hundreds of thousands of combinations of blood groups that you put together to get each individual profile. And working in the transfusion centre, you get all of those really difficult cases to identify antibodies or identify cell types that the rest of the routine hospitals can't do. So that was really fascinating. And I was able to do on call where you'd get called in the middle of the night and they'd need some blood for a really sick patient, or maybe they were doing a liver transplant, or one of the scariest ones was a really small baby who was having a really bad reaction, antibody reaction as they were born. They were having reaction to their mother's and that mother's antibodies were reacting with the baby. And so solving those problems was just really exciting. And transfusion medicine is the one laboratory science where if you get it wrong that day, your patient can die that day. So I really enjoyed that, and it was really exciting. So that sort of led me to be really passionate about helping patients. But also the day-to-day, when it wasn't on call, that was quite routine. And you can probably already tell that I like to talk. So being stuck in a sort of lab all day with my hands in a fume hood doing sort of bottling of reagents and things, it kind of was not necessarily checking all the boxes of what I wanted to do. And so I was reading a magazine one lunchtime, and it said, there was one article in the back and it said, so you want to be a CRA. And I had no idea what a CRA was, so I went off and looked into it. And it turned out that as I researched it, this became almost my perfect job. So I stumbled into this. It wasn't something I planned. It wasn't something I knew about going through school. But a CRA, as we said in the introduction, is a clinical research associate. And it's an entry-level role into clinical trials. And so as you're a CRA, you get to use all the science. You have to know about which sort of medicine or pharmaceutic you're developing, how it interacts in the body, what are the side effects, what's its profile in terms of pharmacokinetics. You have to be able to talk about the indication the drug is designed for. But so you need to use all your science, but you also have to go out there and visit clinical sites. And so a clinical site is where the physician is going to use the drug that you are developing and test it in patients. So this role is great. You get to go out there, you're meeting with the physicians, you're helping them understand how to run the protocol. Protocols are a word you hear a lot in clinical trials. So the protocol is the process you're going to follow, and it's very strict because you need to collect data that shows that that product is safe and effective, and you have to have parameters around how you're testing it. So here's this role where you get to travel, you get to meet people, you get to talk about science, and you get to help get drugs to patients and to market. Because back in the 1960s, drugs used to come to market quite quickly. And there was a product called thalidomide, which came to the market. And if you sort of ever heard of thalidomide, it caused birth defects. It caused children of the mothers who were taking it. And it was actually prescribed for morning sickness, but it caused the mothers to have children who'd be born without limbs. Back in those days, there wasn't testing for drug products. So the thalidomide events are actually what caused a lot of the regulations, the rules that we have around drug development today. And that's what we're trying to make sure that doesn't happen again anymore, and that we have all of those rules in place. So the role that I had when I was a CRA involved making sure that the clinical trials are run properly, so we get the right information to register a safe drug that also works. One of the first drugs I was able to work on was, we'd call it almost a lollipop or a sucker, and it was called Actic in the US. It's actually an opioid. It's a transmucosal opioid treatment. And actually what that's for is for breakthrough pain in cancer patients, and also for burns dressing patients, children who've had horrible burns. If you want to just give them enough drug to be able to change their dressings without allowing them to sort of become overdosed or drowsy on the opioids. So this product was a great one because actually we did this, it had been registered in the US, but we wanted to get it a license in Europe so that European patients could benefit. And so we ran a clinical trial across sites in the UK where cancer patients were able to use this drug, and the data was able to show that this drug worked, and therefore it became to market. And through that work, you know that a lot of patients who were suffering from different types of cancer breakthrough pain were able to get a treatment. And that's what's really satisfying in these roles. So there's varying roles of responsibility that you can go through after being a CRA. You move into project management, where you help run the clinical trials across the globe. And while I was doing that, I actually went off and did my master's. So my master's I did whilst I was working. It was very focused on the roles that I had, and actually really helped me enable to progress my career. But again, the key sort of role there was, it was something I was really interested and passionate in. So that's how I ended up landing where I did and following that path.
Thank you. And question number 2 is, can you briefly describe what drug development is and how clinical trials fit into the process?
Yeah, so I think I already started to touch upon this. So drug development has many phases, but you can start at the top level with research and development, and you hear people talk about R&D. And the easiest way to compartmentalize those is research is everything you do before you give it to a human. And development is everything you do when you're treating humans and patients. So research, you're identifying a pathway or an indication or a mechanism of action. And then you're deciding, how do you target that pathway? How are you going to interfere with it? Are you going to use an antibody? Are you going to use a small molecule? Or one of the many other platforms? There's many, at least 15 different approaches to in treatments at the moment that we have, that makes it really exciting. But you decide what pathway you're going to interfere with, how you're going to deliver the drug, what does the drug look like, and you test it in vitro, in cell panels, and then you do move on to some sort of early preclinical work. And at this stage, you're looking to say, has it got the right binding mechanism? Is it at the target I thought it would be? And you have to put together what we call a package of information, and it's the new drug application, an IND, and you put together information, you send it to the FDA, and you say, I now know enough about this drug that I want to give it to humans. And so you move out of research into development. And so development consists really of 3 phases. And I think it's very timely to be talking about this because we're all waiting for a COVID vaccine, and everybody's saying, why is it going to take so long to get a COVID vaccine? Well, they're racing through the R part, so they're identifying the right antibody, how to sort of build it up to scale. And then you go into the development side, which is 3 phases. So you have phase 1, phase 2, and phase 3. So the first phase 1 study is normally just a few patients or a few people you give it to. If it's oncology, it is normally a patient, because oncology drugs are normally much more potentially severe. The risk-benefit profile, you're prepared to take more side effects in oncology for an effect versus, you know, something for sort of pain or arthritis or something that would be different. So sometimes it's in patients, and then other times it's going to be in healthy volunteers. And often it's a healthy male volunteer because we don't know the effects. Remember, I spoke about thalidomide at the beginning. Often it's easier to pay young, fit, healthy males, and you do normally get paid for being in a phase 1 study, to take the drug. And often it's a single dose, and you're looking to see, did it work in the way I wanted it to in the blood? So can I measure it in the blood? Was it in the right places that I wanted it to bind? And also, were there any side effects that I wasn't expecting? And so that's phase 1. Once you've got that data, you start to go on to phase 2. So phase 2 is normally always in patients. And in phase 2, you're looking to say, what's the right dose where I get a treatment effect? I don't want to give too much, but I also want to be in that window where it's just the right amount. Because obviously it costs money to make drugs, so we want to be giving the right amount to get the right effect, without giving too much or too little that we don't actually get the effect we need. So phase 2 is normally all about dose finding. And probably you go from probably having around 10 to 15 patients in a traditional phase 1 study, maybe up to 30, up to many more, around sort of 50 to 60 in a traditional phase 2 study. Obviously there are numbers either side of that. But phase 3, which is our final phase, and I heard today on the radio that one of the vaccines for COVID is just about to start phase 3 in July. And that's where you start to give it to a lot more people. You know the dose that works here, and you're going to say, does this work or not? And so all of these phases that I've spoken about, the clinical trials, that's the clinical trial operations aspects that I've always been passionate about. So phase 3, you're normally going to say, I'm going to give half of my people a placebo. So they're getting, we don't know what they're getting. They're getting either an injection, but there's no active product in it, or they're going to get the active product. And we measure it. We look for response. We look for effect. We look for side effects. Or sometimes in indications, you may already have a pretty good, what we call standard of care. So you may use standard of care as one of your arms, and then the new treatment as the other arm, and you're comparing the difference. And all of the agencies to approve a drug will expect a certain different statistical significance. So for those of you interested in statistics, it's everywhere in clinical trials. There's lots of statistics. We do power calculations, we look at Kaplan-Meier curves. I mean, the data coming out of trials leads lots of interpretation, not just thinking about data and the results, but data in terms of how you choose your sites, etc. So that's really the clinical trial process. And there's so much within it that makes it really exciting and diverse.
What does a typical day in your profession look like?
So I was thinking about this, and I actually don't think there is a typical day. I think it's the sort of profession that if you like waking up and completing a to-do list, it's probably not the place for you, because my to-do list changes every day, but normally there's all sorts of problem solving to do. It's about being able to sort of maneuver around the issues to help people resolve the issues, to be able to sort of manage the curveballs. There's also staff management, like with many roles. And then there's lots of sort of interactions with people. So you'll be interacting with the clinical sites. That's where the physicians are treating the patients, with the nurses who are helping you collect all this data. Because what I haven't mentioned is that when the patients are at site, we're doing lots of measurements. We're measuring blood pressure, we're collecting side effects. And all of that's entered into, it's all electronic these days. When I started, it was on paper. But today you enter it into a system and it's anonymized. So if you're a patient in a clinical trial, the pharmaceutical company never knows your name. We just know a number, and that's how we're able to keep it anonymized and do data protection. But we're talking to the clinical sites. There's ethics committees. You can't give a drug to a patient or to a human without having had it reviewed by an ethics committee. So we're talking to ethics committees. We're also sort of tracking our goals and then meeting with senior staff. We're helping design the protocols. And then you have to stay current with the science of whatever the indication or the product is that you're working on. So there may be conferences and training to keep up to date with that. So it's depending on which space you're in clinical trials, because there's lots of roles within the space I've been discussing. You can become a statistician, there's data management, there's project management, there's working in the suppliers. But I would say most of the roles need science and people. You have to like sort of interacting. And so that would be really where a typical day sort of goes.
And that builds on the next question about, would you describe your job as requiring more business background or more scientific background?
Yeah, so I think if you're in the traditional clinical trial space, such as my roles were at Amgen and before Amgen, then certainly the routine entry is a Bachelor of Science degree, and the ability to understand the scientific pathways, the biology of what you're studying. In my current role today, as the VP of Business Operations, that's much less so. It does help to understand the science, because you need to be able to understand what people are speaking to around you. But I think people can come into this role from very different backgrounds. What's needed in my current role is more experience and the ability to, again, problem solve and to do many different things in one day.
Question number 5 is, what do you enjoy most and what do you dislike most about your job?
So I think that I've been really lucky, and I've always enjoyed this industry, and I've enjoyed what I do. Sometimes in the past, the travel could be a lot. Certainly, I enjoyed it when I was younger, and when I used to cover the UK and travel, that was great. I used to go and visit all the physicians. It becomes global, and then you have time zones, and you can end up starting your day talking to Europe at 6 a.m. and finishing your day at 9 p.m. talking to China or Australia. So I think time zones are the things I like the least, because of that sort of requirement to always be on. And your email never stops growing when you're in clinical trials or in any global roles, because there's always somebody online who needs something. So that's probably the biggest downside. But what I really love is that people in clinical trials or in drug development tend to be passionate. They're there because they want to help patients. They're there because they really believe in the science of what they're doing, and that can make a difference. And having the opportunity to sort of discover something that's going to help many people is just a really something that's a real privilege to be able to do. And what I like is when you put a team of people together like that, the power of what I call the group IQ is really significant. And so now what we need to do as an industry is get faster and leaner. We need to start to use technology. I think clinical trials have fallen behind and drug development in using technology as much as we could have done compared to companies like areas like banking. That the technology available in AI and things has probably not been leveraged as much as it could be. Sometimes because the regulations, it's so strictly regulated that the regulations take a while to catch up when it's an organisation such as the FDA or the European Medicine Agencies. So I think that using the group IQ to solve those issues and keep the costs of drug development down is something that is going to be a really great challenge that I will sort of continue to enjoy as we move forward.
Thank you. What is the process for managing large-scale clinical trials while at Amgen, and what kinds of people will you interact with?
Yeah, so I think I've answered this a little bit, but Amgen used a model called a functional service provider. So there are sort of three traditional ways you can run clinical trials. So the first one is called fully outsourced. So within the industry, there's the pharmaceutical biotech companies doing one part of the work, and there's clinical sites, the investigators and the site networks that are treating the patients. And there's sort of a group of companies called contract research organizations. And these are sort of groups of people, they don't own the product, but you hire either the company or you hire their people to run the clinical trials. So if you're a small company, you probably fully outsource your studies, which means you have one person who's going to be the project manager in your company, but you hire all of the other skills at the other company at what we call the CRO. And so there's so many CROs out there. That's the entry level role a lot of people come into, because these sort of organizations, you don't always need to have people sort of monitoring the trial or doing the data management. If you're a small company, you need to hire it for that time that you require that sort of expertise. So fully outsourcing, some of the large companies do it, but a lot of smaller companies do it. Amgen run a functional service provider model where they use their own processes and their own systems, but they hire people in and they hire the people management piece, so they don't have that sort of component, because to some extent contract research organizations, they're experts in providing site monitors, the CRAs and the data managers. So it allows the pharmaceutical companies to focus on what their expertise is, which is the science of the drug development, and lets the operations happen at that CRO. So that's another model. And then there are some companies that still have all of the roles in-house. That allows you to be less flexible if you're sort of pipeline, which is what we call all the products. Sometimes pipelines are a bit like sort of snakes that eat and then they don't eat. And so you have a lot of work in one phase and it goes like this. So if you have all your own staff, it's difficult to manage numbers, whereas if they're outsourced, they can move on and off of the products at the other company and come onto yours when you need them. So there's fully outsourced, functionally outsourced, and then in-house. They're the sort of three main models that can be run in clinical trials. Interactions, I think I've already said, they can be the physicians, they can be the medical advisors in-house. Sometimes you're talking with what we call operations, which are the people manufacturing the drugs. We need to have compliant medications that we can dose that have been manufactured from different places. So there's just a lot of people on a daily basis.
Thank you. And our last question before we can have student questions is, how does a biotech company start? And what is your role in nuco-oncology therapeutics?
Yeah, so for a biotech company to start, you have to have what I would tag an asset. So you have to have either the ability to sort of target a pathway, you have to know that your science is going to lead to a treatment. You don't have to have the treatment because it's going to take, I've sort of spoken about that long process to get there, but you have to know that if I bind this antibody to this site, this is the implication to the pathway, and have some proof around it. So, and then the other piece is important, is you have to own your intellectual property, because if you don't own your IP, which is the intellectual property, then somebody else can come in and do the same thing and it's not got any value. So that's kind of the starting point. And then you need somebody to fund you. So you need somebody to say, I believe in your product, and here's so many millions, or sort of how much you need to set off the company. And then it's really about company startup. And so my role today is to sort of do everything that's not in the lab. So if I'm doing my work well, our scientists can be at the bench and doing the design, because that's where the value is. And I'm helping run payroll and IT and getting the lab set up and making sure that we have all the financials in place. So very different, completely pivoted from my Amgen role, but leveraging all of that sort of the skill set, I think, of how to interact and how to navigate and how to problem solve. And that's what's kept it really exciting, because it's still about getting new products to help patients. And at the moment it's oncology, so that's sort of really exciting to be doing.
And now we have time for 2 more questions. If anyone else would like to ask a question in the chat box. Our question is, how busy and demanding is your work schedule?
Yes, so I think it's as demanding and busy as you'd like it to be. So I think a lot of roles these days, you could work 24/7. I think it's about understanding what the priorities are, and really being able to walk away, especially when our offices are now in the spare room or in our sort of down the corridor. But it's definitely busy and it's definitely demanding. But I think that it's something that you can get behind and be quite passionate about. So it's never a burden to do.
And our next question from Rachel is, how do updated and new scientific advancements, stem cell regeneration, gene editing, etc., affect treatments that biotech companies are creating?
Yeah, so they're really highly competitive. So all of those spaces, I think almost every product that's been developed has some sort of biomarkers or genetic components that we're looking for, because personalized medicine is certainly going to be part of the future. Understanding which patients will respond, which pathways are involved in their sort of indication. There's lots at the moment about treatments such as CRISPR or CAR T cells, which is where you're specifically doing editing of genes to remove the oncogene and being able to replace it either with the patient's own genes and growing some new sort of so it's not like a transplant, but it's sort of almost a replacement that you can do. And so all of these spaces we'll see come through and continue to develop. Very competitive, a great sort of buzz at the moment across the industry. And I think that each time new information comes out, new companies are coming through because they've already seen something in a specific pathway that they can build on. So it makes it very competitive, but that's only good, because at the end of the day, all of us are going to, whether you work for Amgen or wherever, you still are a patient or a sort of daughter or a son or a mother, and everybody needs new products. At the end of the day, if I needed a treatment or my family does, I want to know it's safe and effective. So the more we bring forward, the better it is for everyone.
The next question is, do you know main components of the drugs supposed to suppress or kill the coronavirus that are in development?
No, I think there's so many of them. I'm actually really excited. I'd like us to understand more about who is it that's going to be really ill versus those people who actually are asymptomatic carriers. And I think that's one of the spaces where I know people are looking into that, because it would be great if we knew who needs the vaccine first. It's this population, because their biomarkers tell us that they're going to be more likely to be severely ill. Other than just the background indication, we know that the people with the different sort of background indications, such as high blood pressure or cardiovascular disease, are more likely to become ill. But I think there are other things in there, because you get perfectly healthy people being brought down and then others that aren't. And even things like, I started off talking about blood groups. Sort of recent research has started to say that if you're an O blood group, you're less likely to be severely impacted by corona than if you're an A or a B. So how is that happening? Because it's about the cell surface. It's about how the virus is accessing the cell surface. So I think we'll have treatments and vaccines. And again, the more that come through, the better the chance of us finding something that works. But I'm quite convinced that biomarkers have got a role to play in this for the future.
And if we don't have any more questions, I think we can wrap it up now. Thank you so much, Ms. Carter, for joining us today, and thank you everyone who watched.
Thank you.